Makes sense? For below our max, we can absorb all of it, right? We put this much, we're still okay, we can absorb all of it. We can absorb more, but we can absorb whatever we put in. If I put in this much, then I can only absorb this much food. The rest of this goes out as feces. We lose all that nutrients, right? That's waste. That's food we could have used as nutrients later on. So there's always a match of how much our small intestines can absorb and how much chyme we put in there. Because if you put too much, we're wasting some of that food. That food's just going to move by, it's not going to get absorbed. But if you don't put enough, then we're kind of wasting potential, right? If we're putting a sub-amount, then we're missing out on all this absorptive potential, essentially. So there's always this match between how much chyme we're putting into the small intestines and how much we can absorb. And how much we can absorb can vary, but there's just this kind of basic control that we're kind of metering out how much chyme we can absorb. And that kind of happens at the stomach-intestinal kind of junction here. And so, like I said, the main job of small intestines is to absorb food. And what's one way the small intestines can maximize absorption? By increasing surface area, right? If you increase the surface area, you increase the area that you could interact with food, absorb nutrients, and so small intestines try to do its best to maximize its surface area. And biology does that by making folds. The more folds you make, the more surface area you have to a given volume. And so this is a cross-section of the of small intestines, and we can see that we have a whole bunch of folding here. And this folding, each of these kind of buds that come out, or these papillae that come out, these are called, each one is called a villus, or villi for plural. And so these are just protrusions from the mucosal cells that are going to stick out to the lumen so you can increase the surface area. But it doesn't stop there. So this is one villus. Even if you take one of those cells on the villus, so a cell on the protrusion, it has its own protrusions. So protrusions on top of protrusions, there's inceptions of protrusions. And so this layer is called the brush border. And so once again, this is the basolateral side. This is the apical side. Sorry, I'm just switching colors here. And so what happens is when absorption occurs, it takes molecules from here, move it past the mucosal layer, and then into the capillaries or the lymphatic system, depending on if it's fat or some other nutrient. And so that's the main job of the intestines, is to transport molecules from the lumen to the basolateral side of the cell, so it can then eventually diffuse into the capillaries or into these structures called lacteals, which we'll talk about probably next week. So once in these capillaries, they're now officially, once the nutrients get into the capillaries, they're now officially part of the bloodstream, part of the circulatory system, and the first destination of the circulatory system when it's nutrients is the liver. So like I said, food is going to go down through these veins. So you can think of this, let me draw it in another color. This is, you can say this is the intestines. And then and so these nutrients are going to go into the liver. And they go through a vein called the hepatic portal vein.
They're going through the hepatic portal vein?
Via the hepatic portal vein, they go into the liver. So, this is the second place where we have seen a portal vein system. What was the first place we saw a portal vein system? We haven't gone over the kidneys yet. Yeah, hypothalamus and the pituitary gland. This is the second one because we have one capillary. Good, that's a horrible color. Let me change colors. Much better. One capillary system, and here's your second capillary system inside the liver. So you have a hepatic portal vein system here that takes our nutrients that we've collected in our small intestines and our large intestine, and then they send it to the liver. Why do we have a portal vein system there? Why don't we just dump all our nutrients into the bloodstream and have it eventually make it to the liver? Because look at this, like the blood eventually does make it to the liver. Why don't we just, take a shortcut? Why is it not efficient? What do you mean efficient?
Well, we've got all the nutrients concentrated in one vein.
Mm-hmm.
Why distribute it through the body?
Yeah, right. If you put it into the systemic system, you then have diluted all your nutrients, and now what you're feeding your liver is a much more diluted set of nutrients to process. There's another reason, though. It involves one of our challenges. Filtration of what, though? Yeah, so there's this pathogen issue. There's this toxin problem, right? Some things can make it past the stomach acid into the small intestine. Some things can even get absorbed by the mucosal layer, and so if they make it into our bloodstream, do we want to expose that toxin to the rest of our body? No. So we send it to the liver, and the liver then processes it out, maybe destroys it, does something to it, and then it will then send it back into the system. And so this is really kind of like think of the processing plant. It should be like this. And so that's why we have this portal vein system. So we can also concentrate these nutrients, so they all get funneled to one place and not get diffused around our whole, many liters of blood, but also to act as like a first barrier of protection before it gets to the rest of our body. Because once it's in the rest of our body, it's a lot harder to destroy. It's much harder. This is why people have colds for so long, because it takes forever for your immune system to find all the bacteria and destroy them. Yes?
Around those capillaries of the intestines, that's also going to play a role with the lymphatic system too.
Yeah, that's what this green stuff is. That green stuff is part of the lymphatics. These are lymphatic capillaries. This is what we call a lacteal. Hopefully you remember that from anatomy. But we'll talk more about this next time when we talk about fat absorption. Lacteals are important when it comes to absorbing fats, and so we'll talk about that next week. So what I'm going to do now is I'm going to go back and do some review, and so I've sorry. I'm going to go over the baroreceptor reflex. So this is going back to cardiovascular physiology. And so, just as a review, we can say that the mean arterial pressure is correlated with the cardiac output times the total peripheral resistance. So, what does this mean? Cardiac output is how much liters of blood are we circulating per given unit time. So liters per minute. Total peripheral resistance is how much resistance is there to the flow, right? And so the mean arterial pressure is literally how much hydrostatic pressure are our arteries feeling. Basically, how much. This is analogous to how much stretch are you experiencing in those arteries, right? If there's more pressure, your arteries are going to get stretched out, right? If there's less pressure, they're not going to get as stretched out, right? The same idea is putting water into a balloon. The more water you put into a balloon, more stretched out it gets, the more pressure is there is in that balloon. So, there's this baroreceptor reflex that helps maintain the blood pressure we have in our circulatory system, right? We want this Goldilocks phenomenon where we want just the right amount of blood pressure, right? Too high, vessels start bursting, right? Too low, blood flow won't overcome the resistance to flow, and it won't perfuse, it won't get into organs, it won't reach to your arms and limbs because it just can't overcome the resistance. So we need some middle ground of blood pressure to achieve proper flow without destroying anything. And so that's what the baroreceptor reflex does. It help maintain a certain level of blood pressure. And so here is our left ventricle. And comes out into our aorta. And then it eventually branches out into arterioles. So I'm going to give the exact same example I gave in the cardiovascular lecture. Let's say because of some local control, one of these arterioles vasoconstricted. Let's say there's blood loss there, some weird, pathology, whatever. This closed up. Its radius decreased. What does that mean for the resistance of that arteriole? It increases. What does that mean for the total peripheral resistance? If that arterial resistance increased, what does that mean for the total peripheral